Welcome back to the second session in this education day. Just to make an announcement, in the evening we have the evening event. The address is at the homepage and now it's also in the app, but you have to restart the app to find the address. It's Wollzeile 27A, 1010 Vienna. Okay, with that, we start now the second session, and it's my pleasure to introduce Hendrix as the first speaker of this session. And she is talking about sources, pre-analytics, and preparation.
So thank you for the kind introduction. And of course, it's also my pleasure to be here and be able to discuss with you on the different sources that we use to analyze extracellular vesicles, the pre-analytics that can have an impact, and which preparation methods that we can use. Now, isolation, purification, fractionation, separation are all different types of terms that are actually commonly used in literature to describe the process of preparing extracellular vesicles from their source. So in this presentation, I will focus to another level of heterogeneity, which associates to the methods that we use to analyze and characterize extracellular vesicles. So heterogeneity does not only have a biological origin in terms of the different biogenesis pathways or cells that are releasing extracellular vesicles, but also from the different methods. Now, in general, when for your particular study goal, it is important to prepare extracellular vesicles from a source, there is a particular decision cascade that can be ideal to implement. So first of all, it's of course important to select the type of source you want to work with, but also to explore the composition of the source, because this will be informative to design the preparation protocol that will allow you to separate extracellular vesicles from other materials that are also present in the source. And after that, of course, it's crucial in a third step to evaluate the EV preparation that you obtained. Why? Because then you will be able to understand which different types of extracellular vesicles and particles were retrieved with the preparation protocol that you designed. So let's kick off with selecting the source and exploring its composition. So commonly used sources include urine, cell culture supernatant, and blood. Now, if we try to understand the composition of these sources, it's important to recognize that there are biofluid or source-specific particles that are residing in these biofluids. So, for example, in urine, there are very abundant Tam-Horsfall protein complexes. In cell culture, there can be an abundant presence of cell debris, but also ribonucleoprotein complexes, for example. And in blood, most abundant type of particles that are present there are lipoprotein particles. Besides that, there are also some more general types of particles and components that reside in all these sources, such as proteins and protein aggregates, but also nucleic acids. And of course, besides that, there are different types of extracellular vesicle subpopulations which we aim to analyze and study. But it is important to recognize that not only extracellular vesicles are part of the source, but that there are different types of particles. And when you study these particles more into detail in terms of their biophysical or biochemical characteristics, It can be very informative to decide how you will set up the preparation protocol starting from a particular source. So here we have the different types of particles indicating according to their density and size. And then you can observe that extracellular vesicles fit into a very complex mixture of different types of particles and at some point overlap within their biophysical characteristics with other types of particles. So this overview is crucial to understand which types of protocols or which type of methods you can use and would be optimal in your particular condition to separate extracellular vesicles from these particles. Now, once you have selected the source and identified the composition, another important consideration are the pre-analytics. How will you collect the source? How will you process it? And how will you store it until you're able to isolate and further prepare extracellular vesicles from it? So there are multiple pre-analytical factors, both for urine, cell culture, supernatant, and blood, that have been identified. Some of them are source-specific, and others are, of course, more general and apply to all the different sources. I will not go into detail into those that are listed here. It's also not a complete list, but what is important is that ISAF has multiple source-specific task forces that are really challenged with the task to try to identify pre-analytics that can have an impact on the downstream analysis of extracellular vesicles. So one example I want to highlight here is in the case of blood. So for blood, it all starts already from which vein will you start to collect the blood sample? Which needle will you use to extract the blood? Which blood collection tube? How will you process the blood sample? Will you work with blood plasma or serum? And to exemplify that it can have an impact, I want to highlight here a study where we analyzed the 10 different blood collection tubes where we collected blood from a single donor and compared the downstream quality of the blood sample, but also the downstream extracellular vesicles that we were able to retrieve from these different types of blood samples extracted with the different blood collection tubes. So we evaluated the stability or the impact of the blood collection tube on the stability of extracellular vesicles, the overall quality control on the sample obtained, factors including such as hemolysis, but also activation of platelets, the different blood EV subtypes that were contributed, as well as a downstream omics analysis on the EVs that we prepared from these samples. Now, all these results we plotted together in a correlation analysis where you can actually observe that it does matter which blood collection tube you implement to your downstream analysis. So, for example, some really still are able to activate platelets, have abundant concerns related to hemolysis, different types of blood cell-derived EVs that are contributed, but also an effect on the downstream omics profile of these extracellular vesicles. So when even before you start to collect the source, there are different pre-analytics that you should consider, because once you decide to work with a particular blood collection tube, it's best that you stick to the protocol and to these selected pre-analytics, because otherwise it can downstream generate variability in your results. So we selected the source, we understand the composition, we know that pre-analytics can have a downstream impact, but how do we then design and implement the preparation protocol? Well, there are different methods that are routinely used to prepare extracellular vesicles from a biofluid. And I already highlighted different terms with the opening slide, but for example, some methods are commonly used to concentrate the sample. Reduce, for example, the amount of volume we're working with, which is particularly of interest when you're working with urine and cell culture supernatant. These methods typically, for example, are based upon size, such as ultrafiltration or tangential flow filtration that allow to reduce the volume of the source. This is what we call the concentration step. In the next step, we can aim to enrich extracellular vesicles, meaning that we try to deplete other types of particles from the source and resulting in the enrichment of extracellular vesicles. Also for this, different types of biophysical and biochemical characteristics can be exploited. Most commonly, these are exploiting size, density, or surface characteristics such as the charge or the presence of surface antigens. What is actually important to consider, however, is that depending on the choice of method that you make, you will have a differential enrichment in different types of particles, which may also have an effect on the downstream analysis. Indeed, let's have a more in-depth look to the different types of particles and how they react when we implement a particular biophysical characteristic. So for example, during an enrichment step where we have different types of particles, including extracellular vesicles, can be lipoprotein particles, protein aggregates, and so on. If we separate them on size, you will be able to deplete the smallest particles, for example, from extracellular vesicles, but there will still be a co-enrichment with particles that overlap in size with extracellular vesicles. The same applies for density-based methods and surface-based methods. So if we really want to separate extracellular vesicles from these other types of particles, most commonly we would need a combination of methods, which can be a combination of different size, density, or surface-based methods, but ideally a combination of different methods that exploit complementary biophysical or biochemical characteristics. This allows you to separate, in the end, extracellular vesicles from other types of particles. But you also have to recognize that when you implement such a strategy, that it will also result in the fractionation of extracellular vesicle subpopulations. We know from the morning session that EVs differ and are very heterogeneous in their size. So if you apply a size-based method and you select particles based upon a particular size cutoff, you also have to recognize that you make a selection of EVs in terms of size and that you're studying a subpopulation of extracellular vesicles that have this particular size. Consequently, this results in the observation that the more methods you combine, the more specific your preparation will be. And with specific, we mean pure. So the more enriched it will be in extracellular vesicles and the more depleted in other types of particles. Nevertheless, it also means that we will lose part of the subpopulations of extracellular vesicles and that we have a reduction in efficiency. Now, this is a theoretical background, but I also want to highlight this and confirm this making use of a couple of examples. So as a first example, here an experiment where we collected conditioned medium from breast cancer cells and we processed the conditioned medium downstream with three types of methods. One density-based method, a size-based method, and then also a volume-excluding polymer. And from these obtained extracellular vesicle preparations, we analyzed the downstream RNA composition. What we could observe is that the identification of the RNA cargo content significantly differed based upon the method that we used to prepare the extracellular vesicles. Now, indeed, when we did a further in-depth analysis, we observed that it was only the density-based method that was able to deplete Argonaute-2 containing ribonucleoprotein complexes from the extracellular vesicles. So, which were co-isolated in the other two methods, indicating that the RNA profile that we observed here with the size-based and the volume-excluding polymer was confounded by Argonaute-2 ribonucleoprotein complexes that are also known to stabilize and carry RNA. In a second example, I want to highlight that also the choice of method not only impacts the analysis of cargo composition, but also function. So here we started from an experiment where we collected conditioned medium from cancer-associated fibroblasts using density gradient centrifugation, so a density-based method. and two size-based methods, differential ultracentrifugation and size exclusion chromatography. Here we evaluated the capacity of these extracellular vesicle preparations to induce the maturation of monocyte-derived dendritic cells. What we could actually observe is that it was mainly the EV preparations that we obtained by the density-based method that were able to induce the maturation of these dendritic cells. Looking more into depth for explaining this observation, we actually observed that it was the density-based method that was able to efficiently deplete soluble immune suppressive cytokines that were still present in the preparation obtained with differential ultracentrifugation and size exclusion chromatography. So again, the experimental setup will here highlight the functional or decide on the functional effect of the preparation that you obtained. Now this is even for very simple biofluid sources, cell culture supernatant, but what if we look more into depth into a complex biofluid such as blood plasma? So remember, in blood plasma, the most common type of particles are lipoprotein particles. And if we again look in this schematic based upon size and density, we observe that high density lipoprotein particles are smaller compared to extracellular vesicles, but they overlap in density. Lower density lipoprotein particles overlap in size but differ in density, indicating that if we would combine both, we would likely be able to separate extracellular vesicles from the lipoprotein particles. So we set up a SEC in combination with density gradient centrifugation and analyze the different types of fractions. Now let's first have a look to the different fractions that we obtained with the density gradient centrifugation. And we analyzed the lower density fractions and the higher density fractions, where we observed on the top fractions abundant presence of lipoprotein particles, and in the higher density fractions, the extracellular vesicles. But what would now happen if we do a downstream proteomic analysis to identify the protein composition of these extracellular vesicles after a size-based method only versus the combination with the density gradient? Well, again, here we observe that there is a significant difference in the protein profile that we obtained using the combination of a density gradient versus a size-based method only. And what is of interest here is if we implement the size-based method only, the protein composition that we identify associates more to the protein composition of the top fraction of the density gradient containing the lipoprotein particles and does not reflect the true extracellular vesicle composition that we identified in the higher density fraction. So it indicates, these examples indicate that it's really important to well reflect on how to design the preparation protocol, because it will have an effect on the downstream analysis, both when it's cargo or functional analysis. Now, there are different types of protocols. We have established our protocols in the lab. There are other research groups that use more or less similar setups, but publish different protocols. And there is not one way good to move forward. It really depends on what the aim of the study is and what you want to achieve with the downstream analysis. It's just important to be aware that these factors can really affect the downstream analysis. Indeed, if we take a look into literature, we notice that there is a lot of methodological heterogeneity. So here we map the different types of protocols that were reported in over 1,200 publications for experimental parameters related to the preparation protocol and the characterization. And those 1,200 publications report over 1,000 unique protocols to analyze extracellular vesicles from these different sources. And almost 20% of these studies did not characterize the downstream, the EV preparation that was obtained. So there is a lot of methodological heterogeneity. You could think it is bad, but actually I'm very happy that we have this methodological heterogeneity, because there are still many hidden secrets about extracellular vesicles and the different types of biofluids that we are trying to analyze. Think, for example, about protocols that help to identify and discover the exomeres, the supermeres. And in our particular case, by implementing a particular protocol that we set up in the lab, we were able to be the first group to discover the presence of bacterial extracellular vesicles in the systemic circulation of patients that were suffering from an intestinal barrier dysfunction, including, for example, HIV patients, inflammatory bowel disease patients, and cancer patients treated with chemotherapy or radiotherapy. So yes, methodological heterogeneity is very important to understand the full complexity of extracellular vesicles in different sources. But we need tools to cope with this methodological heterogeneity to ensure that we can also adhere to scientific reproducibility, which is fundamental when we are studying and reporting on any type of EVs from any type of source for any type of application. So tools have been established by the community during the past couple of years. One of the most important tools are, of course, the MySEF guidelines. So here I highlighted the MySEF 2018 and 2023. For me, one of the most important parts of the MySEF guidelines really associates to the need of multiple complementary methods that allow you to reveal the true identity of an extracellular vesicle preparation. It really helps you to assess what is the specificity or how much am I enriching for EVs and depleting other types of particles. How much EVs am I recovering? What's the effect on efficiency? And also, what is the integrity of the EV preparation I still obtain? And for that, there are different types of complementary characterization methods that are crucial to implement. A second tool that was developed is the EVTrack knowledge base, which is a freely available online toolset to stimulate researchers towards transparent reporting. So here the aim is really to provide a framework to help you on how to transparently report your experiments in a material and method section for a manuscript. And a third tool that, of course, has been developed over the past couple of years are reference materials. Different types of reference materials have entered the field, but these are really crucial and fundamental to help us set up preparation protocols, understand the impact of pre-analytics, and also help us to set up the characterization methods that we use to analyze these extracellular vesicles. So as a take-home message, I want to conclude that it's very challenging to study and analyze extracellular vesicles in biofluids. And unfortunately, no, there is not one gold standard method that we can all implement, because the protocol that you need to implement really depends on the aim of the study. What do you want to show, and what do you want to use the application for, and what is the source? Nevertheless, if you implement a stepwise approach of trying to understand what is the source I'm working with, what is the composition, then look which protocols would be able to combine to complete these, to separate these different types of particles, combined with an in-depth evaluation of the EV preparation will at least help you to understand what you're working with and make appropriate conclusions for your analysis. Thank you for your attention, and I'm happy to take questions.
Okay, thank you very much, Anne, for this highly educative talk. I'm sure there are a lot of questions. Yeah, please.
For this very revealing presentation. I have a question on a very basic nature. You showed this beautiful trade-off between specificity and efficiency, between separation versus pre-concentration. So my question related to the, is it a fundamental trade-off, or is it just specific to a technique that's being used, ultrafiltration and centrifugation approaches? Or if this deadlock between high specificity, low efficiency, and vice versa can be resolved by bringing to forefront the different techniques. And related to this question is on the functionality characterization, when you mentioned a different proteome of a different subpopulation of cells. Is presumably most of these studies done well after separation or pre-concentration. So if it is done in situ or during these steps, would you observe the same result or very different?
Yeah, so thank you for these questions. So first of all, related to efficiency and specificity, it's something that we notice with all the analysis. I know I did not really go into depth, but for example, if we only implement the size-based method, your recovery would still be, well, depending on how you set it up, could still be up to 80 to 90%. But it's when you want to deplete as many non-EV particles as possible, you have to narrow down the fractions you select, and then recovery using the same method can go down to 50 to 60%. So it really all depends on where you put your own trade-off between what you consider is a need for the level of purity. If you need a very high purity, you will have to be more selective in the fractions that you select, and of course, then it will have an effect on efficiency consequently. So for now, with all the methods that we have explored, we can highlight that it's impossible for the moment, it doesn't exist, and I think this is really the gold mine for the EV research field. If you could find a method that is and highly specific and highly efficient, that would be the fantastic end goal. And related to the functional and the proteome analysis, there are multiple aspects to consider here. So related to the function, in our experience, the density-based methods deliver the most specific EV preparation, but here we basically observe that you also still further deplete some soluble cytokines. The question is it part of a soft corona that is adhering to the EVs, and how is this needed for the functionality? That's of course a different topic, but it just further demonstrates that depending on the method, you may deplete or enrich for a particular corona that can further impact the cargo composition or the functional analysis.
Thank you.
Okay, we have time for one short question.
Hello, thank you very much for your talk. I actually have two short technical questions. I was wondering what was the rationale of using of 0.5% of the EV depleted serum in the cell culture media. Why this value? And was it commercially viable or was it prepared by you?
Yeah. So that's two critical questions, because that's one of the pre-analytical factors when working with cell culture supernatant. So we used with fecal cell calf serum, which was depleted for extracellular vesicles through an ultracentrifugation protocol. We use 0.5% because we use ultrafiltration to concentrate the high amount of cell culture medium to a lower volume, and if you have a high abundance of serum, it will block the filter and not allow you to concentrate, and not 0% because it then had an effect, a negative effect on the cells. So to ensure their viability and still enable us to perform the protocol, we took the in-between road of having a reduced serum concentration.
Okay, thank you. And then the second question, what I saw from your TEM imaging, negative staining, is it safe to say that for the lipoproteins, they don't collapse during the drying in comparison to the EVs in general?
No, they don't collapse. They do collapse. I'm looking to the electron microscopy specialist. They do collapse. Sorry. Okay. Yes.
So it's just image dependent then?
But probably the high density lipoprotein particles are too small to collapse. Oh, they do collapse.
Okay, thank you.
Okay, very last question from Evie.
Thank you very much, Wolf. So very quick, immunoisolation. What are your considerations there?
For? I missed the first.
Immunoisolation of EVs.
Immunoisolation.
Yeah, in terms of specificity and efficiency, and maybe you could spend very quick and few words on that.
Yeah, so for immune capture, so based upon surface antigens, It can be a very specific one. You, of course, need to know the target protein, and you will also enrich for subpopulations of extracellular vesicles, which is the same suffer we have when we select on size and density. So in our particular case, we usually start from a bulk analysis, not based on a particular protein, because we want to discover novel surface-bound biomarkers, but that would be the next step, that we use these surface-bound biomarkers for immune capture, because it can be much faster, of course. Yeah.
Okay, and thank you very much again.
Okay, I'm happy to introduce our next speaker, who is Tijana Jovanovic-Talisman. She's coming from City of Hope, and she's going to talk to us today about quantification and single EV analysis. Thanks, Tijana.
Thank you for your very kind introduction. It's really a pleasure to be here. And I think that today we kind of heard a common theme, and that is that extracellular vesicles and particles are quite heterogeneous. There is challenge in this heterogeneity, but there is also wealth of information. But to get there, we really need to use multiple techniques often, good standards, good protocols, and report things properly. And MySEF guidelines certainly help with that. So, you know, we have this beautiful set of different types of particles and vesicles that are released from cells. And vesicles are great, right? There is so much information. We can use them to understand how cells functions, what happens in pathological conditions. We can use them in biomedicine, right? But they're heterogeneous in content, size, and origin. And this is just one single cell. If we kind of think about complexity of the systems and we make things more complex, one cell, right, that there is this beautiful islet that is composed of different types of cells, organs or chips, and then obviously we strive going to mouse or animal models and going to humans. We have more and more complexity. So where does that complexity comes from, right? We have different types of particles and vesicles, as we discussed. But in our human system, right, or in animals, we have these additional vesicles from the microbiome. You just heard Hendrix comment, and earlier also Clotilde mentioned bacterial vesicles, those are Niel vesicles, fungal, right? There is a lot going on, and we need to find those vesicles that we care about. And, you know, if you consider biofluids, as you heard, there is a complexity in types of particles that we can detect. Those are lipoproteins that are really abundant, and those particles or vesicles that we care about. But what really happens is that different types of vesicles and particles have overlapping properties. And, you know, I put just few really great references here for you that I got these numbers from. So beautiful complexity and a lot of information. And in terms of what happens in our plasma or other biofluids, yes, we do have vesicles that we care about, often from different tissue types or disease, but there is complexity in types of cells also that come from different organs, right? And not all organs are represented equally in biofluids, and in different biofluids, obviously, we have different proportions. So when we start our investigation, often we have to start with these bulk measurements because they give wealth of information. Those omics technologies really can go deep. They give us an average vesicle, right? Something that we like to make as a picture of what we think is there. But really what happens in the sample is that we have all these lovely, sometimes a little funky looking vesicles that often are those that we care about. So there is complexity, and in that complexity, we find vesicles that we care about or that are important for us. So there is heterogeneity, right? And we sometimes need to understand these subpopulations because we can then really say, okay, if I can separate these, perhaps I have better dynamic range. I can tell more about my sample. I wanted to point one more thing. sometimes we need to go deeper, right? Sometimes not only that I need to use single EV method, but I need to use single molecule method, right? Sometimes I care how many molecules of a certain kind I have in the vesicle. Sometimes I care about if my molecules are randomly organized in the vesicle, such as you can see for WGA. So it's all over the vesicle. Sometimes they cluster, right? You can see EGFR being clustered in the vesicle. So there is also additional level, and that is a single molecule level. So I try to summarize some of the methods that we use for the single EV analysis. And you know, all these tables in a little bit colored by, you know, our eyes. So I apologize for my eyes. This is how I see it, right? So there is no best method. This is really something I want to emphasize. Methods are often complementary, and there is the best method for my application. So I need to ask, what do I care about, and then answer what method should I use. So when we start with these measurements, you know, we often do nanoparticle tracking analysis, right? Even as the first start to kind of approximate our concentrations or sizes. And there's this lovely camera that measures motion of particles and assigns the size. And we can say something more about also charge or fluorescence content if we have these filters. And also we can use tunable resistive pulse sensing. So two compartments, there is a little pore in between, and we measure kind of the volume of vesicles passing through the pore, and that gives us this resistive pulse that we can kind of provide as a measurement. And then we can go further, right? On top of these biophysical or multiparametric approaches, we can use surface-enhanced Raman scattering that really gives a wealth of information of biochemical composition of the sample, of the environment of the vesicle. And of course, we can go further and talk about other multiparametric methods. So methods that will also give us not only size and shape, but also the content. One of those is single particle interferometric reflectance imaging sensors. Mouthful, really, but it's a really lovely technique that tells us something about the size of the vesicles using interference microscopy, but it also uses affinity labeling, so it can tell us about particular content or something about molecules that we care about. Are they there, and are they abundant or not? And of course, nanoflow cytometry is one of the most advanced techniques that is multiparametric in this field. And it's probably one of those techniques that has the best regulations of what you should report, how should you do your measurements, what standards should you do. So there is really a wonderful set of different articles that can tell you how to do this properly, and different also labels that you can use. So this is a really nice method that is really pushing the field forward. What I really love about this field is that the EV field pushes the technology, and then technology really kind of explains the state of the art in the EV field. And of course, we can use AFM that can tell us something about not only the shape and size of the vesicles, especially in the Z direction, but this is kind of one of those unique techniques that can tell us something else, and that's the stiffness of vesicles. And of course, all of us here in this room know about TEM and cryo-EM, two techniques that use electron beam that is high energy, passes through a thin layer of the sample, either whole mount or plunge freezes the sample, and really produces these beautiful images of vesicles that tell so much about morphology and size, right? Really great effects. And we can combine that with immuno labeling to learn are those particular markers we care about there. And of course, there is super resolution microscopy, something that I do a lot in my lab, that can tell us about shape, size, and also tell us something about the concentration of particular cargo that we care about. And this is really a colored view of the table that I was mentioning in the beginning. You know, what can we learn? Again, not a perfect technique, but often complementary techniques and techniques that we can really use for our questions. But what are other considerations, right? Do I need to isolate my vesicles, or I can use crude biofluids or crude samples? You know, how much of that sample I have, sometimes that will determine what I will use. Often, I need to care about analytical sensitivity. Someone brings me a little tube, I need to know, well, you know, how concentrated are those EVs? You know, do I need to detect the smallest one or the biggest ones? What is the range that I can detect with my techniques? Can I detect something that has one molecule inside, right? So those are all considerations for you that you need to think about. Cost and instrument availability. As simple as it sounds, sometimes this is really important. If I want to do a particular measurement, I actually need to worry about this. And of course, I care if my students are really upset when I tell them to do something, because it's sometimes unreasonable to spend hours and hours of so many samples. So hands-on time and user-friendliness, really important when you consider a technique. And when I was trying to think about what I can tell you about advances and challenging of particular technique, obviously, you know, I go back to something that I use a lot in my lab, and that's microscopy. So there is really a wealth of information classical, conventional fluorescence microscopy can give us, but vesicles are small, right? And they have these heterogeneous sizes, and because of this, we actually have to think, if those two vesicles are really close together, you know, maybe I cannot resolve them, and that's when really this resolution matters. So sometimes we have to use super resolution microscopy methods, and often fluorescence microscopy. So I'm going to tell you a little bit about single molecule localization microscopy techniques. And in a way, this is really like watching fireworks in a lab. And my students tell me it's watching fireworks, but for way too long. And I'll tell you why. This is a single vesicle, right? And I'm giving you kind of a little movie of how we image vesicles. Molecules are activated, they turn on and off several times, and then they really kind of compose these pictures because we kind of add frames together, right? So it takes quite a bit of time to image that one vesicle that we care about. And it's never boring. It's always this molecular firework that gets me excited. But what does that mean? How can I explain this? Well, if you like art, you probably remember those beautiful pointillistic paintings, those dots that really combined show a whole image. So think about, in one time point you have one subset of molecules, another time point you have a different subsets, and so on. You don't see the full picture unless you combine them, right? And what you have to worry, where is the challenge here? Well, what if my question is how many molecules I have in my mustache, right? This is a beautiful mustache of, from George Seurat's picture of pointillistic painting. Remember, molecules turn on and off. I have localization. So if I don't count properly, my pointillistic mustache of Seurat becomes surrealistic mustache of Dali, right? So I have to be careful how I count. I should not overcount. So that's a challenge. So we have to continuously use standards, use good techniques to report things properly, right? To do this right. So what can I do then? Well, I can really use this technique for a really beautiful set of information, but I need to know my limits, right? So we talked about this in the beginning. So what is really that lowest concentration that I can use? And this dot right here at the very bottom, for those abundant molecules, the tetraspane CD9, CD9, CC3, and 81, that's 0.1 microliters of plasma. Not much, right? So it's lovely. But remember that when I am looking for those rare vesicles, things are a little more challenging. And then I can apply immunocapture and look at subpopulations. And you heard Clotilde this morning, sometimes we care what is the population of molecules on the surface. So if you can isolate them and say something about their size, their shape, their molecular content, we will have the wealth of information. Again, what are the challenges? Well, remember my students telling me that this fireworks is great but takes too long, right? So it's the time management, right? Often specialized equipment, need for specialized labels. My collaborator says, hey, I have the GFP containing vesicles, can you image them? Sorry, right, I need to use additional things. Things get complicated. So really, what the future perspectives, the way I see them, are kind of adapting already available techniques for our field. One of those is super resolution radial fluctuations, or SURF, and other fluorescent fluctuation techniques. So we kind of get enhancement by doing really lovely processing that's already available in the field. But that also means that we do need to advance data analysis, right? Now we start seeing these lovely vesicles that are close together. And how are we going to do that? Well, machine learning is one great tool that I wanted to mention. And we always have to use techniques that are complementary, right? We need to correlate our data. We really need to be sure that what we are seeing is real and it's there. So with that, thank you for your attention. I'm happy to answer any questions you might have, and I hope that I told you that this, again, heterogeneity has its challenges, but wealth of information as well can be harnessed from it.
Okay. Yes, please.
Thank you so much. It was a great presentation. Quick question regarding the challenges with the EV count. So over the last 10 years, I used NTA, NanoView, NanoFCM, you name it, ZetaView. And I can tell you, if you have one sample counted right now, you're going to get four numbers. And you count it 3 times, you're going to have 12 numbers. So my question is, what is your favorite way of counting the vesicles? Let's say you want to do assay, you need 1 billion. What is your way of or multiple, or what are the combination of methods you like to use to have an accurate number to count EVs?
So there is no right answer for this, but there is kind of some things that I always consider. NTA doesn't really consider those smallest particles, right? And doesn't distinguish between aggregates and EVs. So that's a challenge. When I do super resolution microscopy and I have affinity labeling on top, I really can know that I have a particular kind of particles, but I am looking at subpopulation often, right? So I can tell you that you have to look at the range, right? Most of the techniques have a particular range of EVs they count or not. And remember, we go from 30-ish nanometers or even 20-ish if we are considering exomeres and suprameres as non-vesicular extracellular particles to 10 micrometers. So the range is really what I am trying to say, because if you're measuring particular subpopulation, maybe NTA is a great technique for you, but if you have something that is really heterogeneous, then I would like to use something that has bigger range and bigger sensitivity. So for me, I find things quite reproducible with super resolution measurements, but again, there are other challenges that come with that technique.
So what is your favorite machine or technique to count?
Oh, for me, absolutely super resolution, but again, it's colored by my experiences, right? And I often have things that are heterogeneous. But if I have to do something fast, right, then it's the other answer. If I have a subpopulation, it's the other answer. So it's not one size fits all because of that, because we all have different samples, and each of us have to consider different points of it. Do I need to purify EVs, right, or isolate EVs or not? That's another thing, yeah.
Thank you so much.
Of course.
Okay, just to stay on time, I'm going to end the discussion there. So let's please thank Tijana again.
Okay, before we move on to the next session, just a remark. to all of you, the talks will be recorded and afterwards uploaded into the YouTube channel of ISAF, so just that you know. And with that, we continue with the next topic, imaging. So we welcome Guillaume van Niel.
Yeah, thank you for the invitation. So today I'd like to give you an introduction about these very promising approaches to study EV, but also to give you a critical eye about how to use imaging techniques. So what we will do, we will just follow the chronological order of the way we've used imaging to study extracellular vesicles, and all started already quite a long time ago with electron microscopy, with the dust from platelets, and little by little we understood better and better how these vesicles were formed and their morphology, up to looking at these vesicles formed directly in vivo. And with the use of techniques that we are usually taking from the cell biology approaches, we could finally look at these EVs in vivo, but still at an organ scale. And we had to wait a bit more time to be able to see single EVs in vivo in various model organisms, such as zebrafish, of course, so to better understand the biology of these vesicles in situ. So if we start with electron microscopy, as we have heard right before, we have different types of electron microscopy. The most useful one is transmission electron microscopy, but you can also use cryo-EM, scanning EM, or atomic force microscopy. Each of these techniques will use a different way to process your samples by conserving their properties or after chemical fixation. Each of them have different resolution. It's always a matter of few nanometers, but for 100 nanometer vesicles, it can be important. You can combine them with labeling, especially with transmission electron microscopy, and you can look at your vesicles either after isolation or in situ. So I will mainly develop the transmission electron microscopy. An electron microscope is like a microscope, but using electron instead of photon. So what you will see is the electron passing through the grid and the shadow of your samples. And to look at this shadow, we need to contrast our sample. And for that, you will mainly use a contrasting agent that will fill in all the grooves that you have on your vesicles and that are dense to electron. That's why you can see your vesicles. Yet, to do this kind of transmission electron microscopy and negative contrast, you need to fix your samples with chemicals. That's why most of your vesicles have this red blood or cup-shaped appearance. This is not the natural appearance of your vesicles. The natural appearance of your vesicles is obtained after snap freezing and vitrification of your samples for cryo-electron microscopy, and you can see that you have a balloon-like appearance. This technique of TEM can be combined with labeling. So if you know your epitope, especially at the surface of your vesicles, you can use antibodies coupled to gold particles, so you can label your protein of interest at the surface and be sure of its association with the surface of the vesicles. As we've heard previously, the electron microscopy is one of the unique methods to really appreciate the morphology of your vesicles. And based on the morphology, you can deduce a lot of things about the vesicles. First, whether they are large or small, what is their appearance, whether they are filled with something that is dense to electron or not. You can also distinguish them from different other particles that are in your supernatant. This is an experiment where we have done. We have isolated vesicles and mixed them with different lipoparticles, HDL, LDL, and VLDL. And you can clearly see, not only by the size, but also by the morphology and the density to electron, that you can distinguish EVs from HDL, LDL, or VLDL. And indeed, lipoparticles can collapse. So you have to be careful about these aggregates to answer the question of Anne. So this methods is an accurate method to look at your vesicles, at their morphology, but also at their size. Of not, during the quiz, there was a mistake in the question. It was not the useful method to measure EVs. It was the most accurate method, but it was not updated. Too bad for me. So then I have to convince you that it is an accurate method to measure the size of the vesicles. This is an example I have taken from the EMBO course that we have entertained for 10 years with Anne Hendrix, Esther Nuteltoon, and Cecilia Lasser. So we have separated and isolated vesicles by optiprobe density gradient before and after CFSC labeling, and measured their size and concentration by NTA. And you can see that you have more or less the same concentration, and you can find back more or less the same concentration and same size by electron microscopy. So you have a correlation between both methods. But if now you take size exclusion chromatography and look at the different fraction, you can see that between fraction, you can have 1 log of difference in terms of numbers of vesicles. But this is not reflected at all by electron microscopy. Why? Because by electron microscopy, you can clearly see what is a particle from what is a vesicles. And this is exactly what we see here. The first fractions contain vesicles, and the other fraction contain vesicles and something else. It can be membrane debris, it can be lipoparticles, it can be something else. So which method is the most reliable? Well, it will first depend on the purity of your prep, and of course, try to combine all kinds of methods to measure the size and the concentration of your samples. The main problem with electron microscopists is that they can show whatever they want to show. And because we choose the field that we want to put on the paper, and this is the kind of picture you will see on each paper, a small cartoon with 1 vesicles, showing that indeed we have vesicles of a good size with a precise labeling of the vesicles. But this is most of the time not the truth. Most of the time you need a wide field to really appreciate what you have in your samples. And most of the time you have a lot of debris, your labeling cannot be very specific, and you may have vesicles of different size. So it is important in your papers that you show wide field, with zoom in, and to do quantification if you want to use electron microscopy. All right, let's go now to the labeling strategies that we can use for, to study EVs. So I will go through these different type of labeling. So each time you can combine them either with a fluorescent reporter or a radioactive reporter or an enzyme like luciferase to better understand the biology of the different subtypes of vesicles that you have in your sample. Of course, you can use that in vivo, and that will be important not only to look at endogenous EV, but also injected EVs. And for that, you will need to choose the right model organism. So the main model organisms are the mouse, the zebrafish so far, and also C. elegans and Drosophila. Each of them has their own advantages, but also their default. For mouse, we lack of resolution, of course. For zebra, it's still a fish and not really appreciated by a lot of people in various fields outside of the EV. And for Drosophila and C. elegans, it's not a vertebrate, so you don't have all the architecture that you need to study the fate of your EVs in vivo. Yet, with these different model organisms, and by labeling your EVs, you will be able to better understand the biogenesis pathway and the release of endogenous EVs in vivo. You can also inject EVs that have been labeled after isolation and then follow them to better understand their fate, their clearance rate, their uptake mechanisms, their interaction with extracellular matrix, all these kind of parameters that will affect notably the pharmacokinetic of vesicles that are used for clinics. So the first way to label a vesicle is simply using antibodies. We have a large repertoire of antibodies, yet there are some pros and cons for using antibodies. The first thing is that you need isolated EVs and to fix EVs most of the time. So to do live imaging in vivo, it's going to be a bit difficult. You need a known epitope, of course, and although it is compatible with most microscopy and that you can apply it on a whole cell, here on an artificial immunological synapse, in situ in the zebrafish, or by using it for super resolution or flow cytometry, you are targeting an epitope and using an antibody. If you are targeting an epitope, it's not always associated with an EV. It might be present in your EV pellet, but not on your EVs. Second, by adding an antibody and a tag, you may double the size of an EV. An antibody is 25 nanometer. If you have one on each side, then your vesicle is not 50 nanometer anymore, it's 100 nanometer. The second type of dyes are lipid dyes that are quite popular, like PKH67. Of note, PKH67 can create missiles, so you can have aggregate of PKH. You can have also other dyes like MEMGLOW that becomes fluorescent only inserted within membranes and that you can use notably to do live cell imaging in vivo, and also dyes that are infrared dyes or just fluorescent protein, palmitoylated, so associated with a membrane. All these dyes are interesting because they are pan-EV dye. You can label all kinds of EV as long as they have a membrane. They have a long half-life, so that can be an advantage, but that can be also a problem, notably in vivo, because you may transfer this dye from one EV to the membrane of the recipient cell. It may also affect membrane organization and EV function, so if you use, for example, MEMGLOW, maybe you will find that you completely affect the targeting and function of your EV in the recipient cell, as shown recently by Clotilde Terry. You can use then luminal dyes. They are inside, so they should not affect EV fate and composition. For example, the CFSC is an interesting dye that becomes fluorescent and impermeable once inside the vesicles, but become fluorescent only if you have esterase. So you need EVs with esterase to be able to see CFSC labeling, and not all EVs contain esterase. You need also, with this kind of dye, to remove unbound fraction by SEC or by ultracentrifugation. So you can use then other fluorescent proteins that are inside the vesicles, just soluble GFP that you express in producing cells, or, for example, like in the work of Vincent Yen, synthenin coupled to GFP. You will be then able to label certain amount of EVs that are big enough to contain enough fluorescent proteins so you can see them by microscopy. The main problem will be the background in the producing cell, and also whether you or not you affect the specificity of the biogenesis of your EV by overexpressing a protein that may interact with sorting mechanism. The last way to label your vesicles is to combine a fluorescent protein with a membrane protein that is enriched in a subpopulation of EVs. This is very interesting. You can combine, for example, luciferase to look at EVs in mice. You can use a more conventional protein like a GFP or mCherry to look at EVs in Drosophila, or pH-sensitive tag, like the one developed by Lisa Weaver or by us in zebrafish, to look at your EVs in vivo. Of course, depending on the dye, you will have different resolution, and you may have also a good specificity to subpopulation, although now I've listened to Clotilde Terry about CD63 on membrane bud vesicles. I'm not sure anymore about the specificity to subpopulation. Again, be careful. Overexpression of a protein that is a transmembrane protein on your vesicles may affect the composition of a vesicle, its targeting, its fate, its function, and its uptake. However, you can still use this imaging strategy to look at the fate of your vesicles in recipient cells from the docking up to the lysosomal degradation. For that, different approaches are possible, and you can use, for example, APEX or confocal imaging to look at the docking of your vesicles on the surface of your cells, proteins that are sensitive to pH, to look at the uptake in endosomes that are acidic, so you will lose the pH-sensitive dye, and then use other types of protein or ligand that are not always fluorescent, but that will induce fluorescence in recipient cells if you have a delivery of the content of the EVs in recipient cells, up to the lysosomal degradation, where your dye might be more resistant to lysosomal degradation than your EVs. So you may use, for example, the resistance of the GFP to lysosomal degradation to look at the degradation of your EVs. This is what has been shown here, for example, in the work of Kirsi-Arila, to look at the budding of vesicles by Clem, or the fate of EVs in macrophages, or inside lysosome thanks to dual labeling of CD63, or using super resolution to better see the vesicles inside recipient cells. All right. Now, the field is also evolving, also with the challenge imposed by the vesicles, and here are some examples on how we can always improve imaging. The first one is one technology developed by Charley, where it combines two types of dye within one construct. One is luciferase, so you can see the vesicles in vivo, and another one is a fluorescent dye, so you can have a precise resolution of your vesicles. You can also combine the different microscopy and do, for example, live cell microscopy combined with electron tomography to have what we call correlative light electron microscopy. And this is this kind of combination that is useful to have a whole view about what's going on in EVs. And we have now also new tagging approach, while the Degron strategy developed by Anne Wieman, where only what is secreted and separated physically from the recipient cells stay fluorescent. So as a take-home message, the first thing is that, again, one size fits all does not exist to report the fate of an EV by imaging. You have to combine everything, the technology, the dye, to better understand what's going on with your EVs. And each imaging technique has pros and cons. A particular reporter should be chosen based on your question. Do I want to see a global effect at an organ scale, the fate of an EV in a given intracellular organelle? And the labeling strategy should always be validated. It's not because you are using a dye with an affinity for a membrane that it goes automatically to membrane. Always check by super resolution or electron microscopy that indeed your dye is on your EV. And finally, the choice of the microscopy you will use will depend first on the facility that are present on your institute, but also on the specific research question. If you do, for example, like me, cell biology, you need live imaging with a good resolution. If you want to do more clinics, then you will go it does not obey. And that's the same also for the model organisms that you're going to use. So the most important message here, keep in mind, when you look at a dye, it's not an EV, it's a dye. And you may be surprised about what you see afterwards. And with that, I'd like to thank my lab, and I'm ready for questions. Thank you.
Okay, thank you very much, Guillaume. Yeah, questions, please. Okay, maybe I can start.
Yes,
you very nicely talked about the different in vivo models. I was wondering if you use, for example, zebrafish and the worm, if you compare these. Are there different challenges in imaging of EVs when you compare different in vivo models?
So if you compare C. elegans and zebrafish, what is common and very useful is their transparency. You need transparency to see something, and this is not what we have in mice. So, of course, this can be useful. Then, well, we do fundamental science, but we want to go toward clinics for most of the time, especially if we need money. So you need to show that it can be useful for clinics and for humans. So the closer you are to the human architecture, the better it is. And that's why with C. elegans, it is an interesting model with a known number of cells, but it's a bit far from human.
Any other questions?
Maybe I can ask a quick one. Do you ever worry about the functional integrity of the EVs after labeling? Like that the label may cause some differences in bioactivity or something else?
Yeah, I'm really concerned by that, especially since my last publication. We have to be cautious, indeed, about that, and maybe it will be good to be able to correlate a function with a labeling and a biodistribution, but always taking in mind that the labeling and the function are maybe two different things, and maybe we have different effects, indeed.
Thank you very much for your great talk. So I have one question. So that some of the EVs have a very high autofluorescence. So that what kind of the dye do you think is the most high signal-to-noise ratio?
So you're talking, you're talking about autofluorescence of EVs?
Yes, so that if you label the EVs, you should tell that that fluorescence is a specific fluorescence, not from the autofluorescence of the EVs.
Mm-hmm. So the first thing I would do is a negative control. I always use a negative control just to be sure that I don't have autofluorescence. And notably in zebrafish, we have a lot of autofluorescence, notably in the green channel. Once you know that, then you will try to combine the right dye with the right spectrum, the good wavelengths of excitability, to be able to distinguish between autofluorescence and what is associated with your EVs.
Okay, to be honest, which kind of the wavelengths is the most nice for eliminate the autofluorescence?
But
the green?
It depends on your model organism. It depends on the resolution. Infrared is excellent for mice and deep imaging. Green channel, while GFP might be the brightest, but we have now new Alexa tags that we can combine also. Most of the autofluorescence is rather in the red for me, so I would be cautious, especially with the extracellular matrix. So I will go to the green and infrared.
Thank you so much.
Okay, thanks very much.
Thank you.
Okay, our next speaker has traveled maybe further than most to get here, all the way from Latrobe. So this is Suresh Mathivanan, and he's going to talk about Omics, just a small topic, I guess.
Yeah. Thanks, Randy. So thanks the organizers and the chairs for putting in a really good Congress, and this being the largest ISEV, I also congratulate the chairs for that. So I'm going to dwell on Omics here. So now we are at a juncture where we want to know what is the cargo that is present in the EVs, which is probably alluding to a certain function, as well as if you're interested in biomarkers, you're trying to identify a certain cargo or certain set of cargoes that could be discriminative in terms of your biomarker status, as well as you're also into a stage where you want to know what these EVs are doing onto the recipient cell, and you may complement that with using omic technologies. So when you talk about the EV cargo per se, you can try to identify normally the RNA in it, the proteins in it, the lipids, the metabolites, as well as you can also look at the DNA cargo, which again specifically some people study. The epigenomics field is not well documented, but you can see quite a few methylated DNA fragments which are also present in your EVs, and also when you combine epitranscriptomics, there's probably some methylated as well as some modified RNA in your EVs as well. So we all agree now, based on others' talk and especially Anne's talk here, the EV isolation methodology is critical. So one need to understand what they are isolating. And again, we need to acknowledge that in many cases, we may not be able to isolate the EV population to purity. So hence we are enriching for a certain population, right? So the cargo that you're studying, or once you subject that to any omics analysis, what you get out of it is obviously going to be influenced by your methodology that you used for isolating the EVs. In some methods, that is even more. For example, if you're using proteomics or any lipidomics analysis, your cargo analysis may be even more biased because you have more impurities from proteins and lipids in your isolation protocols than any other chances. But however, you need to understand that your isolation protocol is quite important before you go to any kind of omics analysis. So after that, some of the things that you need to consider that when you're doing any omics analysis, I'm going to focus morely on proteomics here as a omic data set analysis here. But however, when you look at how do you normalize between EV samples, so you can either isolate EVs from one particular cell line or a blood or any bodily fluid and then characterize them, what cargo is present in it, or you can compare between multiple states. When you're comparing between multiple states, you need to have a way of normalizing, and then these are the things which you need to probably take into account. You may need to normalize by the total protein amount between 2 samples or 3 samples. You can also normalize by the number of EVs between the samples. You can also normalize by the volume in some cases where you may not be able to normalize by the total protein or in terms of the number of EVs. And also you can normalize by the number of live cells that you're getting the EVs from, especially when you're trying to do knockout of a protein and then trying to understand what happens to the particular cargo of the EVs after the knockout. You predominantly can rely on the number of cells just to study how much amount of EVs are produced, whether there is any perturbation in the release of EVs after the knockout of a particular protein. This is one such example where when you knock out this E3 ubiquitin ligase net4, we can see there is no change in this TSG101, which is a protein, luminal protein that is present inside the EVs, especially the small EVs. But when you look at Alix, which is also a protein which is present in the small EVs, it is differentially sorted into the EVs upon the knockout of the E3 ubiquitin ligase net4, right? So if I'm trying to load equal amounts of protein, you may miss some of those things, some of these observations. So you need to understand that if you're looking for certain, the questions that you're asking, you may need to normalize accordingly. You also need to understand the surface area of your EVs or your cells that you're working with, because now you may be working with small EVs or large EVs, or you're comparing it to the whole cell lysate, because if you look at the whole cell lysate, you end up getting a lot of cytosolic proteins in other compartments, but the membrane proteins will be less abundant in those preparations. But when you're doing an EV prep, which has a larger membrane compared to your cell, when you look at the size of that, you would relatively get more membrane proteins identified. So in our hands, we can identify 30% membrane proteins in an EV prep versus 10% in your whole cell lysate, right? So it just shows EVs are naturally having more membrane proteins because of the cargo that the amount of protein that you work with. So you need to take into account when you're analyzing any of your data, and also need to look in terms of take into account the surface area as well as when you're doing functional analysis. For example, if you're doing a functional analysis on small EVs and you're putting 10 micrograms of protein, and then you're doing the same thing for a large EV, and then that is probably not the right way to do it, because you are probably looking at one large EV here versus 10 small EVs there. So you need to be mindful about what you're putting in there. And also you need to understand how many EVs one cell secrete to maybe understand or answer these questions even better. And that can be done, again, it is variable to cell, and every cell secretes in a different way. So one of the questions I often get asked in terms of proteomics is how reproducible your EV isolation is. And when it comes to some of these questions, right? So some people come to me and say, I did EV prep and then I did proteomics. I was not able to reproduce the protein list that I'm getting. The reproducibility is under 60% or under 50%. So you need to understand various parameters that you're using or you're isolating your EVs from. One such is the confluency, which I'm sure all of you would understand. If you're isolating EVs from a 50% confluence cells versus those which are 90%, you would get a different cargo there. And same way, if you're using different growth factors, any growth factor, for example, if you treat your cells with insulin or, for example, FCS or any growth factors there, or you starve your cells, you induce stress, or you have mycoplasma contamination, that would also change the EV cargo. So you need to understand that and be mindful about that when you're doing any omics analysis. You also need to validate these the identified cargo, those which you are importantly studying further. For example, if you're identifying 3,000 odd proteins from your EV prep, and then you're associating a particular functional state, let's say an EV is curing cancer, in that case, you may need to understand that these proteins need to be validated, or this RNA need to be validated by orthogonal methods. You not do for all the proteins or all the RNA, but those which are important. Why is it important? Because you need to show that these are in the EV prep, and these are indeed present, and these are reproducible by various methodologies. So when you talk about proteomics, which is what I'm going to spend most of my talk on right now, so you're going to use a mass spectrometer where you may need to spend more time on calibrating and looking at the sensitivity of the instrument, as well as then you have to also spend a lot of time on sample preparation and also data analysis, right? When you talk about mass spec-based proteomics, you have several methods of how you can use your EV prep or your cell prep. The cell prep here is when you treat your cells with and without EVs and want to see what are the pathways that are changing, what are the proteins abundance which are changing, what are the RNA that is changing, right? So in that scenario, if you want to really look at the proteomic changes either in the cargo or even in terms of the cell after EV treatment, you can adopt either a label-free based method, or you can use a label-based method, right? So in the label-based method, you can use several ones. One of them is a SILAC, which is a metabolic labeling. You're trying to grow the cells in heavy isotopes or light isotope media, and then isolate the EVs from that, and then try to do an omics analysis of various states, looking at the absolute quantification of these proteins in your EV preparation. Then you can also do chemical labeling like ICAT, or also you can do enzymatic labeling, but these labeling are after you isolate your EVs. You isolate your EVs, and then you can label your proteins and peptides with these, and then you can try to quantify the proteins in them. In label-free method, you can do quite easily without any labeling methodology, where you can just have 2 or 3 or 4 multiple conditions, and then isolate your EVs, and then subject them to mass spectrometry-based analysis. For sample preparation, you can do multiple methods. In this case, I'm just sharing one of the methods that we use. We have a starting material of 5 to 30, which is very standard in our lab for any proteomics-based analysis of the EVs. Then you can use your loading die as a method to digest your EVs, and then run them on either a 1D gel, or you can directly, if you want to do a in solution-based digestion method, you can opt for a different methodology. You can quantify your protein samples by multiple methods. Again, BCA, micro-BCA, CypherB-based stain as well. Nanodrop can also be used if you're trying to use a quick way-based method, but however, I would say if you want accurate protein identification, you're better off using BCA or micro-BCA, because nanodrop may not be as absolute as you know. So when you do mass spec-based proteomics, the first one, the principle in which it works is you're separating your sample. You can either do electrophoresis 1D or 2D, or you can do a liquid-based chromatography method, separate your samples, and then analyze it by mass spectrometer. So it just fragments your peptides in your mass spec, and then these peptides are then assigned to certain proteins. And then what the mass spec analyzes or measures here is mass by charge of the particles. Meaning in this case, your peptides are being injected, they're ionized, and once they're ionized, they're fragmented, and then the masses of these peptides are analyzed, and then they can be matched back. So in theory, you will have an ion generation in your mass spec, which could be an ESI, electrospray ionization, or it could be a MALDI. And then you have a mass analyzer, which is an ion separation column, which is a quadrupole or an Orbitrap. And then you have an ion detection, which detects your mass over charge. So in terms of your EV preparation, as you can see here, you can take your EV lysate and then try to either use in solution digest, where you can reduce alkylate and digest your samples, and then try to either run in a 1D gel electrophoresis even prior to that, or go with an in solution digest. And then based on that, then after you subject that to a mass spec analysis, once you do that, you can identify the proteins and then go on to a biological state. So one of the things that you need to be mindful, if you're trying to use proteomics and if you're using any of the facility managers, you may need to check with them in terms of the reproducibility of your EV proteomics from the mass spec. How you can go about doing is you can correlate, or you can run correlation between two runs. If you have two biological replicates, just run a correlation between them. If you have a correlation of 0.9 and above, your correlation is pretty good, and then you can go on with your data and then try to analyze that. If not, you can try to calibrate the instrument and then try to rerun your analysis. The basics behind mapping your mass by charge, because your instrument, your mass spec, only measures mass by charge, and from mass by charge, how are you getting your proteins, right? So for that, you will have several search engines, which then theoretically what they do is they have an experimental based analysis as well as a theoretical one. In the theoretical one, the peptides are fragmented from a protein after, let's say, if you're using an enzyme called trypsin, and then trypsin normally cleaves at the C terminus of arginine or lysine. So you create theoretical peptides ending with R or K, and then try to analyze the MS-MS spectra for each one of them, and then you compare your experimental MS-MS spectra and your theoretical MS spectra, match the best, and then score them, and whichever scores the highest, you will identify that as your protein of choice, right? So this is one question I get often is, what if I don't have a very good database? So this is based on the assumption that if you're working with a human or a mouse organism, you easily can isolate your EVs and you can match that to a database. But if you're working with an organism that do not have a very good database or a genome in itself, how do you go about it, right? So you can try to complement that with proteogenomics or even transcriptomics. If you have transcriptomic data, try to translate that into a proteomic database and then research it. If you have a genomic data, you can predict the ORFs or open reading frames from your genome and then try to create an MS database and then try to search that as well. But one thing you need to be aware of this proteogenomic analysis is this has a high false discovery rate. If you create, because obviously you're going to create a database that may not be existing in real time, because you are predicting what proteins could be coming from a particular genome. In such scenario, you will have a lot of false discovery rate. You need to be mindful, and hence you need to have a stringent data analysis platform for you to identify real hits from the analysis. This is another question I get often, where people always ask me about the RNA versus proteins, and I I did a EV prep and I identified a lot of RNA. I did a cell-based analysis. I identified a lot of RNA, but I'm not able to identify a lot of proteins. So you may need to be careful about this in general is, again, when you do RNA, you can amplify the RNAs, and even little amount of RNA can be amplified, and then you can try to analyze and identify them. But the protein biochemist cannot amplify the proteins per se. So you're working with very little amount of proteins. The low abundant proteins are normally not identified unless and until you enrich for those proteins in your mass spec preparation. Hence, when you do a cell-based analysis, you can identify 18 to 19,000 RNA, but the proteins is normally, let's say, 4,000 to 5,000, unless and until you do more enrichment for identifying more proteins. Same ways for the EVs, you'll identify only less proteins because there are quite a few which are low abundant, and you may not be identifying a lot of them. And one other thing you may need to bear in mind is, here is one data where you're looking at resistant and parental cell lines of colorectal cancer for 5-fluorouracil. And you can see the P is the parental cell and the R is the resistant cells. You can see this protein called nestin, which is present only in the resistant cell but not in the parental cells. But when you do mass spec analysis, we did not find it. So here it is black and white, but in mass spec, it is not founded. If you have not found any protein in your mass spec, it doesn't mean it's not there, right? So there's multiple reasons why it is not there. Your antibody may work. Just go ahead and throw your antibodies that you have on your prep. Just because mass spec didn't find it, it doesn't mean it's not there. In SILAC-based proteomics, again, you can use it quite a lot in many other analyses. Again, as I said, these are stable isotope labeling of amino acids in cell culture. You can use it to quantify between EVs. You can also put it on your recipient cells and then look in terms of what proteins are changing in that. So one other way is you can isolate EVs from, let's say, your light and heavy cells, take the EVs and incubate that into your recipient cell, and then see what proteins are being transferred. And this can be played out quite nicely in a lot of systems, including mice as well. And then you can study what proteins are physically transferred from a recipient cell, from the EVs to another recipient cell. I'll skip this part. You can also do SILAC mice proteomics, where you can grow the mice in a SILAC diet for quite a generation, and then try to isolate the EVs or inject EVs into them and study in terms of where are these EVs biodistributed going, and then what are the changes they cause to phenotypically in various tissues per se. And then you can study that even without SILAC mice. If you're not having so much money, you can probably have 2 mice states, label the EVs with, again, the SILAC, and then heavy labeled EVs where all the proteins or the amino acids are heavily labeled. You inject them into a mice and then dissect the various organs and study whether these EVs are going in. I should also mention that you need to have more protein for you to do these kind of analysis. You may need to inject the EVs maybe for at least 7 days continuously to see an effect. But if you're doing intravenously, probably do it for 2 weeks to see an effect as well, because you're administering very low dosage of EVs. With that, probably I'm just going to skip these slides. This is one other slide, one other question which I get often, is how do I identify more proteins in plasma EVs? Because normally people do a lot of plasma EV analysis. You can enrich for your isolation method. You can use multiple enzymes. You can fractionate the samples. Instead of doing just 5 samples, you can do 20 samples. You can use a longer gradient. You can use multiple ionization methods, as well as you can use multiple search engines. By doing this, you can increase your number of proteins, and also, most importantly, your isolation method is quite important, right? Other common issues with proteomics, you may have keratin contamination, because when you sample preparation, it may have contamination. You can have too much enzymes, which you need to be mindful, and you need to also look at your correlation between that. There's also, if you want to predict the source of your EVs from your omics data sets, there's also a tool that has been developed, and that is in one of the talks. If you're interested, you can go and check that. with that, I'll stop here and thank all of you for your attention. Thank you.
Thank you, Suresh. Any questions? Yeah, please you can make your way to the mic.
Thank you for your talk. So I'm guessing from your talk that you use mass spec. What are your thoughts about SomaScan or Olink for proteomics in EVs?
Sorry, I'm not able to hear that completely.
So my question is, what are your thoughts about using SomaScan or Olink for proteomics in EVs?
Sorry, it was... Okay. So yeah, I think you can use both of them, actually. So it's pretty good for omics analysis in a way. But however, as I mentioned, you need to be careful in terms of what your downstream analysis, or what is your question that you're asking here afterwards, from after the omics in a way.
Okay.
Yeah. Hey, Suresh, thank you for the talk. I had a question. It's more a complaint from the mass spectrometry facility in my new institute. So they are using database like Vesiclepedia, etc. And I try to explain them that most of the data that we have on this database are coming from poorly isolated EVs, and sometimes we don't know exactly whether that's EVs or not. So he was asking me whether there was a way to clean the database or to purge it somehow, to have really a database on which we are sure about the isolation procedure and that we are looking at is strictly EV associated.
So if I understand your question, you're asking in terms of the EV proteomic databases, yeah, which are like Exocarta, Vesiclepedia, and those, yeah?
Yes.
So in such scenario, you can probably separate by the isolation protocol. But however, I agree with you. There's a lot of bad quality data sets out there because, again, as a field, we have evolved what we call as exosomes. Probably 10 years ago, now in Exocarta, we are kind of deleting some of those data sets and then trying to incorporate them. But having said that, it's a very hard thing to do if you're trying to obtain data from publicly available sources to get high quality data, but which you may need to probably annotate it yourself. You may need to handpick some 10 or 15 studies and then try to maybe make a high quality data set if you're really interested, or you may need to produce it yourself from one lab. That's what I would suggest if you want to have a high quality data set.
Thank you.
Thank you for the talk. So I had a question regarding the slide where you showed that when you get proteomics from the cells, you have like 3,000 to 4,000 you find in mass spec, but when you do EVs, you find 2,000, like, less. So is it something to do with the methodology itself, or is it just generally in the EVs we will find less proteins? Because it's ultimately not a cell, right? You are only localizing certain proteins. So what do you think about that?
That's a good question because, again, when you look at the cells, you have a lot of protein content in it, like you say 30 microgram, and even if you put 30 versus 30 with your EVs, you tend to identify lesser proteins here. Because now when you look at the dynamic range of the instrument, it is going to identify the most abundant proteins and then keep on going in that way. When you look at the cells, there's a lot of abundant proteins in there. When you look at EVs, there's quite a few abundant proteins, but not all of them are abundant there. Plus, also, you're looking at a population of EVs here. Not all of them will have protein in it. You may have 30 micrograms as an equal loading amount. But however, as I said, there's a lot of membrane proteins in EVs when you look at the surface area versus the cells. And membrane proteins may not be easily identified by mass spec analysis because they are hydrophobic in nature. The enzymes that you're using in mass spec, the trypsin, and which cleaves at arginine and lysine, these are cleaving at basic amino acids. That's why you have a lot of identification of your cytosolic proteins, but your membrane proteins being hydrophobic, you tend to miss them because of your enzymes that you're choosing. So all these factors contribute to less identification, and that's why you get less proteins there.
Yeah. Thank you.
Okay, unfortunately, we'll have to cut the discussion here. Sorry, just for the timing, but you can find Suresh later. So let's please thank Suresh and the other speakers from this session. Okay, now we have a panel session, so I would invite the members of the panel to please come up on stage, and then we'll have lunch following. Thank you.
Okay, perfect for the announcement. And while we are awaiting the panelists on the stage, we will have some organizational information by our fantastic sharing team, Eva Rude.
Hi, everybody. So before you are enjoying the panel session, I wanted to inform you that lunch will take place upstairs in the first floor, so please head to brasserie and a Schubert room, because there the lunch is upstairs. You might have noticed we have a graphic recording here. So it is in the making. Robert Six, he is really doing a cartoon where we will have all of the essence of this educational day then depicted in this cartoon, which will end up in the evening. And so far it's in the making. And for this evening, please don't miss the educational day event. In the app, we have now inserted the address of the venue. It's Aula of Research. It's the Aula der Wissenschaften. If you don't find it in your app, it's in the website also mentioned in the program schedule. And if you refresh your app, step out once and go back, then you should have the location. So, and that's it. Thank you.
Okay, I was already wondering, it's fantastic that we will have a cartoon. I'm really looking forward to the result. So this was really a lot of information, fantastic tips and tricks that we already received in the morning session. Time to digest before we can enjoy lunch. So that's why we thought it would be a very good idea to now actually give the word to the hands-on scientists, the early career researchers that have to deal every day with all the challenges that we encounter, and they have to deal with it in the lab. So we tried to make this interactive as well. So all the questions that will be fired to the hands-on scientists will also appear again in a Slido. So what we want you to do is to also contribute your answers to each of these questions, because we will also use it to guide a little bit further the discussion. It are open questions, so please only make an answer using a keyword, because we try to extract the most common keyword answers that were indicated by the audience.
So, without further ado, I would like to ask our panel to introduce themselves and what they work on, especially regarding the EVs. Thank you.
In other words, where are you from? Who are you, where are you from, and what do you do?
I can start. I hope you can hear me. My name is Alessia. I am, till recent, was a faculty research associate at Johns Hopkins University. I also a co-founder of Excel. This is a core facility we established in Hopkins. And who I am an engineer first, and I really love scientific instruments. So I basically, in EV field, I played most with every single instrument. In EV biology, what I'm interested in neurodegeneration and neurodevelopment, and also non-vesicular particles, which is like lipoproteins, for example, from the blood. So I work with EVs from the blood and non-vesicular particles as well.
Hi, my name is Claudio Pinheiro. I am a postdoctoral researcher at Ghent University on Hendrix supervision, so she's right over there. My expertise revolves around isolation methods. So throughout my PhD, I studied 13 different types of isolation methods. I developed different types of tools to aid rigor and standardization in the EV field. I characterized a lot of my preparations using 7 different types of characterization methods, which included deep omics to NTAs, etc. And yeah, that's pretty much it.
My name is Mark Bergquist, and I'm a fourth-year PhD student at Gothenburg University at Jan Latval's group. I mainly work with cell lines and cell cultures, and I engineer cells and subsequently their EVs to overexpress certain adhesion molecules to target and treat inflammation.
Hi, I'm Cristóbal Serra Troncoso. I'm from Chile, but I'm working in Belgium. I'm postdoc at KU Leuven. My expertise is about the cell biology, about the control of EV biogenesis and secretion. And recently, I'm working and try to understand what is the autophagy-dependent secretion.
Okay, thank you and welcome to the panelists. So we look forward to the discussion. Here is the QR code for the Slido, so feel welcome to join the session. And then we kick off with the first question. So first of all, we learned a lot, starting from a source that you have to prepare extracellular vesicles for particular downstream applications. So there are multiple methods you can select. So starting from your experience in the lab, what are the most important steps that you consider when you set up a protocol to prepare EVs from your biofluid of interest? Yeah, let's kick off with Claudio.
So I think I will take a bit of inspiration from a lot of the talks that we heard this morning, and I will break it down into 3 main steps. The first step is really understand why you're isolating your vesicles, because that will really assist in deciding exactly what type of isolation method you will use in the future, because that has very important, like we just heard from the previous talk, it has very important decision cascades down the line. It's just a domino effect. Step number 2 is actually understanding really well what is your biofluid and what the biofluid has. So in the talk of Professor Guillaume, we also heard how having lipoprotein particles can actually affect the readout, or for example, even deep omics. So in this case, I would say, make sure that we understand very well what is in the biofluid other than your vesicles. So you will have, of course, vesicles, but you will have several other complementary particles that you will need to either deplete or try to remove in order to be able to actually study your vesicles. And step number 3 is actually understand that indeed there is no one key fits all for all methods. There's pros and cons for each of the method that you will try to use. And with regards to that, the key is actually understanding what these pros and what these cons actually are. And based on your actually informed decision, you will be able to then actually select. So I really like to put it in a way that you need to understand if you want to have a lot of vesicles, but you probably have to sacrifice, like we saw in the talk of Anne, we'll have to sacrifice the specificity and ergo have less amount of, or less purity, let's say, or the other way around. And that really will dictate the selection of the most appropriate methods to your specific study goal.
So Cristobal, as a follow-up, because you're more on the functional side of EV analysis, do you have particular considerations on that point of view, like how do you want to preserve the biomolecular corona, the EV heterogeneity to ensure the functionality is maintained after preparation? So at what level do you see preparation for functional studies on extracellular vesicles?
I'm missing the question a little bit, but I think you always need to think about the limitations that you have in your analysis. And from there, you need to think how the conclusion will come from that limitations. So sometimes will take more time in the analytical analysis of your experiments, but at the end will give you best results. So yeah, that is from my side. And yeah, so to preserve, you need to consider that your sample will be frozen at some moment, and the defrozen will affect the samples. Try to don't defrozen and frozen many, many times. And in my hands, I always prefer to do all analysis as possible from the same samples. So analyze the EV particles or quantify what is inside, and then use that's the same sample for Western blot analysis and yeah, etc.
Yeah. Okay, I think we see many of the comments that were touched upon also coming back on the survey. Speed, high throughput is a particular one that we did not really go into detail, but I think from the current experience, the faster and the more high throughput, the more difficult it also is to have a higher level of specificity. But there are definitely, and I hope you have the time during the week to also visit the different booths from the commercial partners, because there are very nice developments to really try to combine all of this in a single equipment. Okay, thank you. Then we continue to the next question.
Yeah, so next question would be like, so we have multiple characterization methods available, like NTA, electron microscopy, or like single EV analysis. So what would you recommend, like which methods would you combine to get a comprehensive view about your EV preps? So the question is to Markus.
Yeah, I would recommend using at least one method to check for the different EV markers, if that's nanoflow cytometry or Western blot or all the different times that exist, but also one to check the morphology, for example, EM or cryo-EM or something like that. They are very complementary.
What I usually tell people when someone like comes and you're like, you have like basically all the instruments, what should I do? I can suggest you can use them all and never get the results you want and results you expect, because each method is very unique, and the design of your experiment and design how you characterize it depends on scientific questions. What do you want to learn at the end of their study? So basically, if you're just interested to design a biomarker, do you need a single particle analysis? Do you even need to know if you have AVs in your preparation? Because biomarkers is biomarkers, just like I always ask people, did you run just your biofluids itself? Maybe you already have a biomarker. So it's very dependent. But if you really work with AVs and you were not able to find your biomarker, for example, in the biofluid of interest, I always suggest to start with TM if it's available, because it's a kind of quick and easy method to at least see the purity of your AVs, and if you actually have some of the particles you were aiming to isolate.
And Alicia, maybe to follow up, so in terms of characterization methods, some equipment is very dedicated and not always available. So can you elaborate a little bit on how to deal, like if you don't have the methods available, what are the options that you can consider to have a sufficient amount of information that allows you to interpret? Like, is it through collaboration, core facilities? Can you a little bit elaborate on that?
Yes. The first thing I always suggest everyone, just go collaborate. Go on the conferences, just like find, I really want to do, for example, super resolution microscopy. I don't have an instrument. Just go to the poster sessions and say, hey, you did this experiment, can you help me with my experiment? And like people in the V field are very nice, and like most of us, like happy to collaborate and like happy to bring new research in the next level. So just like ask around.
But also Western blot is a very good place to start to check for the different EV markers and even non-EV markers if EM is more difficult for you to access.
Yeah. So different levels of characterization methods that you can use, and even with the very straightforward ones that are available, you can already have a good indication, but always look for collaboration. I think these days research always needs collaboration to be able to do everything. So try to explore your network and see what you can do together with your collaborators to further expand.
Also a little to reconnect to a question from one of the sessions. If you get very varying results from NTA, it's also very good to verify it using other methods. For example, just checking the protein concentration. If you have 10 times more particles from batch to batch using NTA, but the same protein amount, then yeah, something is probably wrong with either the analysis or the sample preparation, for example.
Or just a different person ran NTA at that time?
That's correct.
And what if you only have a limited amount of sample available? What's the minimum you have to do?
Okay, so you have to show that you have vesicles. That's the first step. I think it's important. Step number 2 is to show that you've depleted for the things that you were wanted to deplete. So if you want to show that you are studying actual vesicles and you don't want to, in the end, you want to make a conclusion specifically regarding protein or whatever it is regarding to your vesicles, whatever association it is, you should then definitely be able to demonstrate that you've also depleted the particles or the things that you should have depleted in the first place. I think baseline, that should be what you always need to at least demonstrate. Anything above that is, in my opinion, extra bonus that really demonstrates your hypothesis.
Okay, perfect. This is really a session dedicated for early career researchers. So if you have any burning question in the meantime, please also go to the microphones and we may still have time to join. So then we move to the next question.
So for example, if you're designing an omics experiment and to analyze your EV cargo, so what would you recommend for the newcomers? Like what kind of considerations you would recommend?
I can start with, I had a lot of bad experience with transcriptomics. So I can tell you what not to do when you design the experiment. For example, my recent fail, I did not include the proper controls in my experiment. So what happened, we found a lot of bacterial RNA in my preparation, and I did not know if it's from my sample or because I just contaminated like RNA towards the road. So we had to do an extensive long analysis on my samples to make sure that it was not a sample contamination. So controls for omics experiments. Based on their concentration and how much material you need, it depends on your experiment, your purity of your samples, and what's the final goal of your study.
I would just like to add as well that understand the limitations of the technology as well. Each technology has its limitations, and for example, we just saw the nice talk before on proteomics, and if you have your vesicles, but you also have other contaminants or other EV depleted or other particles that you have there, this is going to overshadow your proteins of interest or your vesicle proteins. And that's actually something that you should take in consideration. Also, I saw a very nice slide on the number of replicates that you should have, because I think it's very useful that you meet together with people that have more experience dealing with omics data, that they can run you through what actually you require to have in order to design a proper experimental setup for an omics data. Whether this being obviously the optimal controls, but how many replicates that you should have in order to, in the end, be able to draw a proper statistical significance conclusion or something like that. That's all things that, in the end, weigh a lot. And these experiments can be quite expensive. And that's all things that you should consider.
Yes, we have one question.
Yeah, hi. This question really relates back to how you characterize your samples. So it seems like pretty frequently you'll do multiple characterizations, and the different methods will conflict with each other. For example, Western blot will say you have one thing, some of this, but none of this. And ExoView will say, actually, it's the reverse. You have none of that and some of this. So I guess my question is, has that happened to you? What did you learn from it in terms of how have you changed how you approach characterization? And if it happens again, like, how do you resolve it?
Yeah, that is, I think, is a little bit eternal question in the field, how to choose the best comparable methods in the field. So in the way that both are going, always you need to take in consideration the limitation of each one. If you can have over conclusion from each one, you need to consider that in the comparison between two methods. But in the way that both the results from parallel methods to characterize CVs are going in the same way, could be the best option from my side. But always thinking on the limitation of each method. For example, if you see in Western blot that you cannot detect the protein in EV's fraction, that don't say that the protein is not secreted. So that you cannot detect, but it doesn't say that it's not secreted. So you need to go in almost, in my point of view, almost 3 parallel methods different to say that you don't detect or is not secreted or is secreted. That is my point of view. Yeah.
Yeah, I just want to say that controls help with that a lot. I don't want to sound like a control freak, but kind of. I want to also say that it never happened to me, but all the time. And it's very, very scary, especially as a newcomer, because you have a lot of expectations when you do a Western blot, and then you're supposed to find the EV markers, but at the end of the day, you spend 2 days on something, and then you have an absolute blank slate. Techniques have its limitations. It's also hands-on a lot of the times involved. That's what I experienced as well. A lot of the times it has something to do, like if you have the proper controls, consider that you have the proper controls and your experimental setup is well designed. And if not, look at the application, how we did everything, and then sometimes that can also lead to a solid conclusion. But of course, that's why we always use orthogonal complementary characterization methods, that in the end, if one says no and the other one says yes, if you have the third one, usually that's like allows to have something in between.
Okay, thank you. And then, so I still want to encourage the early career researchers. This is the time for you to ask questions, so grab the phone. Don't worry that you hold everyone back from lunch, because it's only ready at 12, so that's not a good reason. So in the meantime, we want to check with each of the panelists what is now the exact advice that you would give to new scientists entering the challenging field of extracellular vesicles.
Can you repeat the question? Because yes, sorry.
So, well, it's a question to be addressed by each of you. So what advice would you give to the new scientists that are entering the field of extracellular vesicles, from your experience?
First, start to frustration times, but it's part of the science. Resilience as well, because it's at the beginning can be overwhelmed stuff to prepare EVs, and if your cells are not so secreted, or they don't secrete a lot of EVs, but just take time to analyze your cells, to your models, how it's going, And if you want to analyze a particular protein, try to study the biology of that proteins, because that can affect the result at the end. And yeah, go ahead and try to talk with others that have more experience. Try to follow the talks of ISEF and SNEF, because from that part you can have more ideas and try to cover your gaps on the EV fields. Yeah.
I can say, start with position papers. When I started with in EV field, it was not that many. So I made a lot of mistakes. I had to learn myself. Right now we have like amazing tools available. Start with MyZev. If you haven't heard of MyZev, it's today you have to read it. It's a nice paper. So yeah, or reach out to people who are experienced in the field already, so they can help you design an experiment, and you will avoid a lot of like very common mistakes.
Yeah, just look at MySev and set up a very robust isolation method, because if you cannot show that you have EVs or you don't isolate pure EVs, then all the results in the end, you cannot really show that they're because of the vesicles, it can be because of everything, and then you cannot really publish that data or use that data as EV data. So yeah, a robust isolation method.
Yeah, different than anyone has said. So I really like the invest early time in figuring out the basics. I don't know who placed that, but I think it's super nice, because as someone that is starting in the field, I think the first thing you have to do, and first of all, you've already found the best conference to be at, because this will give you a lot of information based on that. Follow a lot of these early career researchers' questions as well. Do not be afraid of asking questions. Asking questions is part of the job. There are no stupid questions. You can only learn from the questions. If you don't know the answer, now you do once you get the answer. If you did know, now you get confirmation about your answer. So that's actually, you'd never lose in that situation. That's what I like to say to my master students as well. It's a fantastic field to be in. It's sometimes very challenging, but it's worthwhile pursuing it. So don't really believe in the work that you're doing. If you design everything properly and you reached out to other people and you surrounded yourself with a good support, you'll be able to have a very good time in the field.
And I also like the one, do not believe the schematics. I think you can believe, but you can contribute to change them by getting in your knowledge, and also think out of the box. It's not because something is written there that it cannot be different. That's exactly why we need research and why we need joint efforts to fully understand. We learned this morning from Edith's overview, there is plus 20 types of EVs that are released by cells. Probably there are still many, many more. So believe that you can help to change and update the schematics that we find in the books. Final call. Any question from the audience still to be addressed? This is the moment for you. I think all the information from the morning session has been digested then. And then it's time for lunch, but let's first thank the panelists. And then enjoy lunch. You will find it at the end of the hallway, so go back to the registration desk and then go one floor up. Okay. And we have to announce the SNF lunch. Yeah, and maybe, sorry, one final remark related to the lunch. So there is one SNF initiative during the lunch that Cristobal will kindly introduce.
Yes. So at the SNF, we prepare a fun activity for the lunch. So upstairs you can find the tables that are tagged with a particular EV field. So you can sit in the table of each EV field that you like, but it's not for discuss about the science, but it's to know the others that are working in the same field on EVs. So try to be fun on there, and we are working around and chat with you. Thanks.